Hello everyone. In the previous video, I have explained about the first chapter, and now in this video, I'll be doing with the second chapter, that is the sexual reproduction in plants. In this video, I'll be discussing about the flower, a fascinating organ of angiosperms. All of you are very familiar with flowers, which are normally seen around us in different shapes and colors. And the flowers have a very special place in our daily lives. We plant the flowering plants for their aesthetic beauty, and they are used for social, cultural, religious functions. Bees collect nectar from flowers and they convert it into honey, which has medicinal value. The formation of flower occurs as a result of a number of changes in the plant, a number of hormonal as well as structural changes, which leads to differentiation and development of the floral buds, which will turn into a flower. Now, the flowering plants. Flowers are the sites of sexual reproduction. All of you have studied the structure of a flower in detail. Still, I will try to recollect your memory of the structure of a flower. A flower is actually a modified shoot, and a typical flower has 4 whorls which are arranged around the thalamus. The stalk of the flower is called as the pedicel. The outermost whorl is the calyx, which is made of the sepals, and the sepals are protective in function. So the sepals protect the flowers when they are in their bud state. And the second whorl is corolla, which is made of petals which are brightly colored. Some flowers may have fragrance, and these petals help in pollination by attracting insects. The androecium is the third whorl, which is actually made of the stamens, the male reproductive parts, which is called as the microsporophyll. And the gynoecium, which is the fourth and the innermost whorl, containing the pistils or the carpels, which are the female reproductive parts. Now we will study in detail about the male and the female reproductive structures of a flower. First, we will see about the male reproductive part, a typical stamen, which has 2 parts. A bilobed structure, which is the anther, which is attached to a long, slender stalk, which is called as the filament. And the filament is attached to the thalamus, or it may be attached to the petals. The number, the length of stamens vary in different species. In flowers, the stamens show a wide range of variations in their arrangement and attachments. First, we'll study about the arrangements. Filaments of all the stamens are fused together into a small bundle, and the anthers remain free. This kind of a group of anthers is called as monadelphous stamens, arranged in one group, but their stamens are fused together. So this is an example. You can find this kind of monadelphous stamens in hibiscus. In the second kind, that is diadelphous, the anthers are seen in two bundles. The stamens are seen together in two bundles. Here the anthers are free, example P. In flowers, the stamens show a wide range of variations in their arrangements and attachment. In the first case, that is about the arrangements, filaments of all stamens are fused together into a bundle, and the anthers are free in flowers like hibiscus. Such an arrangement is called as monadelphous stamens. Here all the stamens are in one group, one bundle. And in the second kind, that is diadelphous, you will find in case of pea plants, here the stamens are fused to two bundles and the anthers are free. And the third kind of arrangement is polyadelphous. Here the anthers are fused to form more than two bundles. So this kind of polyadelphous condition is seen in lemon. Then there is the fourth kind of arrangement of anthers is syngenesious, and in this the anthers are united together but the filaments are free. And example is a sunflower. Now on the basis of attachment of stamens, we can categorize the flowers, we can categorize the stamens into the epipetals, where the stamens are attached to the petals by their filaments. Example is sunflower. And second kind is epiphyllous. Here the stamens are attached to the perianth. Example is the family Liliaceae. Then the third kind is gynandrous. Here the stamens are attached to the pistils. Example is Calotropis. Each anther is bilobed with each lobe containing two thecae, that is dithecous, having two pollen sacs called the microsporangia. The anther is a tetragonal structure consisting of 4 microsporangia, which the microsporangia give rise to the pollen grains which contain the male gametes. The two lobes of anther are joined by the connective tissue. I will see about the structure of microsporangium. Microsporangium appears near circular in outline, which is surrounded by 4 walls or 4 wall layers: the epidermis, endothecium, middle layer, and the tapetum. So 4 wall layers are there: the epidermis, endothecium, middle layer, and the tapetum. The outer 3 wall layers, that is the epidermis, endothecium, and the middle layer, are protective in function and help in the dehiscence of the anther and release the pollen grains. Tapetum is the innermost, and it nourishes the developing pollen grains. The cells possess dense cytoplasm, and they may have more than one nucleus also. When the anther is young, the sporogenous tissue occupies the center of the microsporangium. Now I'll explain the next stage, that is the microsporogenesis, that is the formation of the male gametes. As the anther develops, the cells of the sporogenous tissue undergo meiotic divisions and form the microspore tetrads. Each cell is called a microspore mother cell or a pollen mother cell. Now, the formation of microspores from the pollen mother cell or the microspore mother cell is called the microsporogenesis, and the cell division here is meiotic divisions, meiosis, and the microspores are arranged in a cluster of 4 cells, the microspore tetrad. As the anthers mature and dry, the microspores get separated and form the pollen grains. The pollen grains are released with the dehiscence of the anther, with the breakage of the anther. The pollen grain is actually the male gametophyte. When you study the pollen grains, you will find a variety of sizes, shapes, colors, and designs of pollen grains. This you will do in your practicals. That time you can clearly see the different size and shapes of the pollen grains. Now we study in detail the structure of pollen grains. Pollen grains are generally spherical, 25 to 50 micrometers in diameter. Each pollen grain is covered by a thick wall having 2 layers. The hard outer layer is called as the exine, and the inner layer is the intine. The hard exine is made of sporopollenin, which is considered as the most resistant biological material known. Sporopollenin. And the sporopollenin enables the pollen grains to withstand unfavorable conditions like high temperature, strong acids or alkalis. And the pollen grains are very well preserved as fossils because of the presence of sporopollenin on their walls. The exine, the outermost layer, has apertures which are called as the germ pores, where the sporopollenin is absent or it is very thin. And the pollen tube emerges from these germ pores when the pollen grain germinates. The inner wall, which is the intine, is thin and continuous layer which is made of cellulose and pectin. Being thin, it gives rise to the pollen tube at the time of the pollen grain germination. Now, a mature pollen grain contains a vegetative cell and a generative cell. 2 cells, a vegetative cell and a generative cell. And the vegetative cell is usually the bigger cell and has an irregularly shaped nucleus. And this vegetative cell is with abundant food reserves. The generative cell is actually very small, which floats in the cytoplasm of the vegetative cell, and it has a dense cytoplasm and a nucleus. The pollen grains are shed at the 2-celled stage in many of the flowering plants. The generative cell divides mitotically and gives rise to 2 male gametes before the pollen grains are shed in many of the plants. Now, the period for which the pollen grains retain the ability to germinate is called as the pollen viability. That is actually the lifespan of the pollen grains. and this pollen viability is variable in different plant species. The pollen grains viability depends on the external temperature as well as humidity. In rice plants and wheat, such as cereals, the pollen grains remain viable for 30 minutes. And it is possible to store the pollen grains for years by storing them in liquid nitrogen at very low temperatures, that is minus 196 degree Celsius. And this way it can be used as pollen banks. The pollen grains viability depends on the outside temperature as well as humidity. In cereals like rice and wheat, the pollen grain remains viable for 30 minutes. And it is possible to store the pollen grains for years by storing it in liquid nitrogen at minus 196 degrees Celsius, and it can be used as pollen banks. Pollen grains are a very rich source of nutrients, so they are used as food supplements as pollen tablets. In this video, I have explained about the male reproductive organ, and in the upcoming video, I'll be discussing about the female reproductive organ in detail. Thank you.